in detail when we will do autoimmune diseases, because we are somewhat short of time. Vasculitis occurs due to immune complex deposits in the blood vessels, and they cause inflammation of the blood vessels. Certain clinical conditions occur like polyarthritis nodosa and IgA vasculitis, SLE, giant cell arthritis. These are all basically arising due to the immune complexes depositing in various blood vessels. So when these immune complexes deposit in the blood vessels, they activate the complement, and the complement in turn activates various mediators of inflammation. Now coming to type 4 or delayed type of hypersensitivity reaction. This is delayed type and it takes some time to establish, and this is not dependent on antibodies. Like I told you before, that type 1, type 2, and type 3 hypersensitivity reactions are mediated by antibodies, while this type of hypersensitivity is mediated by T lymphocytes. And a very interesting thing is that it can be transferred by the cells and not by the serum. Like we saw that in certain conditions, the antibody-mediated types of hypersensitivity can be transferred through serum. But this, because antibodies are found in the serum. But this can be transferred by sensitized T cells, not by serum. So what happens, the antigen combines with the macrophages, and it is then presented to the helper T cells with the antigen, and interleukins are formed, as we saw in the beginning when we are doing these immunological reactions. These macrophages, they are antigen presenting cells, and a reaction occurs against that antigen which is presented to the helper T cells. So one type of type 4 or delayed hypersensitivity is contact hypersensitivity. The other type is known as tuberculoid. Now in contact hypersensitivity, you must have experienced, many of you, that if you use certain chemical like cosmetics or certain soaps, oils, they cause an allergic reaction on the skin. And if that, sometimes that reaction is so severe that it can lead to tissue necrosis of the skin. Many girls are very fond of putting henna or mehndi on their hands and palms, and sometimes a very severe reaction occurs. So this is a very classical example of contact type of dermatitis or contact hypersensitivity. Now what happens, that certain chemicals like nickel, formaldehyde, certain plant materials like poison ivy and poison oak, certain drugs that are applied topically like sulfonamides, neomycins, certain cosmetics and soaps, when they are applied to the skin, the antigens, they get absorbed in the skin and they become a part of the skin cells. Now the body's immune system takes that cell combined with the antigen as a foreign material. And then CD8 plus cytotoxic T cells, they attack the skin cells that have that plant oil or any antigen that, like a cosmetic or any chemical, as a foreign cell, because it has been absorbed on the skin cells. So skin cells are now not recognized as cell. So this contact type of hypersensitivity, because these cytotoxic T cells and CD8 cells are now attacking the skin cells themselves, taking them as foreign material or antigen as a whole, they cause pruritus, vesicular skin rash. Now, in tuberculoid type of sensitivity, certain different types of cells come into action. Here you can see that the memory T cells are also being formed, And with the next exposure to the same antigen, a more severe reaction can occur. And especially people are allergic to certain artificial jewelry also. So this is also a type of contact hypersensitivity. So the small molecules of the chemicals or certain material like nickel or artificial elements, they act as haptens and they enter the skin, they attach to body proteins that act as carrier proteins and they become complete antigens. So cell-mediated hypersensitivity is induced particularly in the skin. And upon re-exposure, contact dermatitis develops more severely. So in the other type is tuberculin type. In the tuberculin type, uh, these CD4 positive helper T cells act. So please remember the difference in contact type of dermatitis or contact hypersensitivity type 4, CD8 plus cytotoxic cells act on the skin cells. While in the other type, which is tuberculin type, CD4 cells come into action. And this type is used for the diagnosis of tuberculosis in a test which is known as tuberculin test. Many of you might have heard about it. So if a person is previously exposed to Mycobacterium tuberculosis, what happens that this small injection, which contains purified protein derivative from Mycobacterium tuberculosis, it is given intradermally. And then we see and wait for 48 to 72 hours. So if the person was previously exposed to Mycobacterium tuberculosis protein or had an infection, a positive skin test will indicate induration and redness at the site where the intradermal injection is given. Why? Because it is a reaction against the antigen which was present in the person's blood. So the positive skin test indicates the infection, but it does not confirm whether it is still there or the infection has been cured. So this is the tuberculosis skin test, very important. It's known as PPD. PPD is the preparation which is given in the skin intradermally. It means it stands for purified protein derivative of the Mycobacterium tuberculosis. So the important thing is that if a person's test was negative before, but later, after some time, he again got this done and the reaction becomes positive, it means that the person has recently infected by tuberculosis. This is the importance. So these are the delayed type hypersensitivity reactions like contact, dermatitis, tuberculin, even granuloma formation. Now we have done all the 4 types of hypersensitivity reactions, type 1, type 2, type 3, and type 4. And I hope that you have some concept about what is the mechanism behind these hypersensitivity reactions. Now, few points for you to understand. There are certain immunological tests that are being done for diagnosis of various diseases. And these are related to the antigen-antibody reactions. One is agglutination test. The antigen is particulate like red blood cells or certain latex particles are coated with the bacteria and or any antigen. Antibody is present in the serum. So when the serum is combined with the antigen-coated RBCs or latex particles, these antibodies combine with the antigen, and these antibodies in the serum, usually IgM, which is a big molecule, they form a lattice or network type, a clumping reaction that can be seen under the microscope. So if the clumping is seen, it means the agglutination is positive, and it means that the person's serum contains antibody against that antigen. So this is how the positive agglutination test appears like. So this agglutination can be seen by microscope. You cannot see it like that without a microscope. Then there are precipitation tests in which the antigen is in a solution form and the antibody cross-links the antigen molecules in variable proportions. So there is a zone of equivalence like this. This antigen is in a solution, and when you add the antibody, the serum, there is a certain zone in which maximum antibodies can combine with maximum antigen. This will form a precipitate which will be seen, it is visible also. It's known as zone of equivalence because antigen and antibodies are in a good proportion, the correct proportion. Wherever the antibodies are more than the antigen or antigen is more than the antibodies, they cannot form a precipitate. So this zone of equivalence is a zone of visible precipitation. This precipitation test can be done on agar plates also. This precipitate will appear on the agar plate. So there are various usages of these agglutination and precipitation tests in the diagnosis of various diseases in the laboratory. Enzyme-linked immunosorbent assay is a method in which again you can see the antigen-antibody complexes, but here an enzyme-linked reaction occurs and it gives a color to the small wells that are present in a tray, and you can confirm the presence of antigen or antibody if the other is known. So same antigen antibody reactions occurs in blood cells. You know that different blood groups contain these antigens, and the other blood group has the antibodies against that antigen. And when these combine, they cause clumping. So if a mismatched transfusion is done, this clumping occurs and these antigen antibody complexes activate